So the next talk is by Dr. Srivatsan Seergazhi. He's an associate professor at Indian Institute of Science, Education and Research, Pune. He has been a senior fellow since September 2016.
Okay. Thank you for introducing me to the audience, and first of all, I'd like to thank the Indian Alliance for giving me this opportunity to showcase our research in this meeting. By training, I'm a chemist, and then over the years, I've started to move into molecular biology, then cell biology, and more. Now we are moving more towards into structural biology. And today, what I'm going to do is that I'm going to talk about our how simple chemistry can be used to understand or address a biological problem. And today, what I'm going to do is that I'm going to briefly talk about our probe development program, wherein we have developed probes to understand mood swings. It's structural swings. It's nothing related to the mood what you're thinking about. It's strictly structural swings of non-canonical nucleic acids. Why we started to embark on this one or use chemistry as a tool to address these problems is the following reasons. As you know, nucleic acid, it doesn't work. How is it? Yeah. Okay. So, as we know that nucleic acid performs several essential cellular functions such as storage and transfer of genetic information, they can do catalysis, they can also regulate gene expression by several mechanisms, right? And in order to perform these functions, nucleic acid use intrinsic conformational dynamics, and then they adopt complex but well-defined architectures, or let us say secondary and tertiary structures, which are made of structural domains such as classical double helical structure and structures which have bulges, hairpin and pseudoknot. And for example, I've given very few sets of secondary and tertiary structures. And also certain G-rich sequence, guanine-rich sequences and cytosine-rich sequences can adopt 4 standard structures called called G quadruplexes and I motifs, and then it is known that these structures can act as regulatory elements. And today I'm going to talk about, today I'm going to talk about developing probes to understand the structural swings in G quadruplexes. Let's see what how G quadruplexes are made of. G quadruplexes are essentially made of 4 guanine residues. They come together through hydrogen bonding interaction, and then they are stabilized by metal ions such as sodium and potassium. And 2 or 3 such tetrads stack over each other and then form G quadruplex structures, and guanine rich sequences that have the potential to form such structures are abundantly present in telomeric repeats which encaps the chromosome. They are present in DNA promoter regions and in untranslated regions of several mRNA, which can potentially cause cancer. And compelling evidence suggests that these structural motifs or structural elements can maintain chromosomes and also regulate proliferation associated genes. And for this reason, these structural motifs have been considered as novel therapeutic targets for developing chemotherapeutic agent. In terms of structure, they show structural polymorphism, and this polymorphism essentially depends on the sequence itself, and then confinement, and it can also depend on the molecular coding effects. And these have been shown to be forming several kind of structures in vitro. For example, human telomeric repeat of a TTA G3 sequence can adopt a parallel structure, anti-parallel structure, hybrid structures, and equivalent RNA essentially forms only one structure, which is a parallel structure. Okay? And other sequences form several of these structures. You don't have to remember these structures. You have to just remember parallel, anti-parallel, and hybrid. That's good enough for you. And my students, they have to remember more than that. Okay? So, several studies have been done over the years, by using NMR, X-ray crystallography techniques and other techniques to understand these structures. But then our current understanding of what topology they adopt inside the cell is not available, and then which topology is responsible for its function. And importantly, if we get to know how to really target these sequences specifically by using small molecule inhibitors, okay, and modulate their function. So, but then these questions cannot be readily answered by currently available tools. For example, antibody based methods are good for detecting the formation of G quadruplex. That itself is a good thing, because until recently only it was proven that G quadruplex can form inside the cell. But what topology it forms, it is not available. And more importantly, these antibody based methods are restricted to fixed cells, so morphological changes and other things can also lead to differences in G quadruplex topology. People have used synthetic cell models as well as molecular coding agent, but then they tend to bias the structure to one of the structures, and for this reason people were thinking that parallel structure which is formed under these conditions, antibody based method as well as in synthetic crowding agent, people thought that the parallel structure is the structure which is essentially could potentially form inside the cell, but that's not the true thing, which you would understand much later when I show the end results. And also most of the tools, chemical probes as well as tools, cannot be effectively implemented in both cell-free and cellular systems, and most importantly existing probes do not discriminate individual G quadruplex structure. That means they don't give clear or distinct signature for individual G quadruplex structures that can be used to understand what structure these sequences can form inside the cell. What I want to say is the following. So I'm going to use a real life example and then get it to you. So what do you think is happening here? What Virat is doing? Exactly, you know, that's a signature, right? He has scored his 100 and he's celebrating his 100. Okay, now let's see this one. So what's happening here? No, not 50. Actually, he has got out, but then he's acknowledging the crowd and he's happy about it because he has scored 100 or something like that. Okay, so you have to watch more cricket or Virat. Okay, so that's the message. Now, what do you think about this situation? He has got out, probably he is not happy with the decision, not that one. He has got out, but then he has not scored as many runs he would have expected. Okay, so that one, you have to see more cricket. Okay, now what's happening? So this is during batting. Okay, now he is fielding. Yeah, he has lost the match. Okay, that's a good thing. So you see one sequence have different emotions and you can clearly have signatures. Okay, if you watch cricket you will know. Okay, and now let us see another celebration. Okay, so Usain Bolt, it's a very typical signature, right? You cannot miss this, and of course you should be knowing, if you again you have to watch football. Okay, so whom do you think is this guy? Cristiano Ronaldo. Yeah, it's a typical gesture. He does like this when he scores a goal. Okay, so these are signatures, but then we don't have clear-cut signatures of G quadruplexes that can be used inside the cell, and this is what is the aim of our research, and this we want to do it in a different fashion. Actually, people use traditional approach of one label one technique or one label one biophysical technique, but then we want to move away from this traditional approach and use an innovative probe design to address this problem, and then we develop multifunctional nucleoside probes which are conformation sensitive. And this is how we develop these probes. We attach, it is known from our own studies as well as others, if you attach heterocyclic rings onto nuclear bases, you can actually expand the conjugation and then you can generate environment sensitive or conformation sensitive nucleoside probes. And in addition, we took advantage of the spectacular, or I would say excellent, anomalous scattering property of selenium atom, which has been very regularly used in protein X-ray crystallography here, and also the high sensitivity and high abundance of 19F label, which could be potentially used for in-cell NMR techniques. So an important aspect of this design strategy is the following. So you can see the fluorophore, the selenium atom, and the NMR label are all in the same electronic system. Why it is important is because the environment each label is sensing could be then correlated by different techniques. For example, you can use fluorescence for real-time analysis, you can use fluorine NMR for in-cell NMR analysis, and selenium for X-ray crystallography. So you would get a comprehensive idea of what's happening in real time, in 3-dimension by X-ray crystallography, and in cells by NMR. So that's the idea. And once you incorporate these molecules into a target oligonucleotide sequence, you can concurrently use fluorescence, NMR, and X-ray crystallography to study all these processes, and then you can nicely correlate. And with this kind of an approach, or design approach, we started to synthesize several of these analogs, because it is not so easy to predict the fluorescence property. So we attached several heterocyclic groups and what is an array of fluorescent nucleoside or multifunction nucleoside we synthesize, and we do a preliminary evaluation and identify the best combination or the best hit molecules, and then we go about and implementing in understanding, nucleic acid structure and function or something like that. So today I'm not going to, overwhelm you with humongous amount of synthetic data and then results, but then I will take one example of a dual app probe, okay, which has a fluorophore and an NMR label to study the structure of human telomeric overhang in real time as well as in cells. During the course of investigation, so we identified this particular fluorescent molecule, which is a benzofuran attached to a uridine ring. I know you are mostly your biologists, but then I thought I can teach a little bit of chemistry. Okay. So what you can see is that this becomes into a fluorescent molecule, and then this happens to be a very good G quadruplex sensor, meaning it shows differences in different G quadruplex structures via changes in fluorescence intensity, lifetime, and quantum yields, and as well as anisotropy. Okay, but then the excitation maximum happens to be in UV region, which is around 340, so it's not so conducible for analysis in cellular environment. So we came up with a simple idea. This happened, simple idea of incorporating a fluorine atom here in the same electronic system, and we did this for the simple reason that it is 100% naturally abundant, and then its chemical shift signals are highly sensitive to the local environment, and one important thing is that the cells don't have fluorine, so you don't have any background NMR signals you can have. Okay. And first of all, what we do is that you don't have to worry about the numbers. I'll just briefly tell you what actually these molecules are up to. So we do preliminary analysis at the nucleoside level to see if these are sensitive to the microenvironment. What we do is that we do fluorescence analysis in solvents of different polarity and viscosity, and then we identify that this molecule is highly sensitive to these polarity changes and viscosity changes, and same is reflected by NMR also. So you get unique signatures for different environments. So it is environment sensitive at the nucleoside level. Then what we do is that we incorporate this molecule into an oligonucleotide sequence, DNA as well as RNA, and we subject them into different neighboring base environment to see how they are responding to changes in the environment. And I'm not going into the details, but then we found out that these are highly sensitive. Based on these results, then what we did is that we wanted to study if these molecules can be used to understand G quadruplex structures or not. For that, we chose human telomeric overhang as the first test system, because it has been very well studied. We can directly compare the structures with the in vitro data and our data and show that the modification does not affect the formation of G quadruplex. In other words, it doesn't affect the structure as well as the stability, but then it reports different G quadruplex structure. The importance of this overhang is that it can form multiple structure. The prominent ones, parallel structure, anti-parallel structure, and mixed hybrid structures in different salt conditions. Okay, but then what structure it adopts inside the cell, it is not very clear. Okay, and one more thing is that we wanted to next decide where we have to position our probe. Yeah. Where we have to position our probe. So you can see from this structure, simple structure is that the tetrad, conformation of tetrad is not much different. It's not too different, okay? But then you see the loop residues, they are quite different in different structures. So we thought that it would be more appropriate to place the probe at the loop residues rather than the tetrad. And also one more thing is that we have seen the crystal structure of the unbound state and the bound state of the G quadruplex structure. You can see the loop residues undergo serious conformational change. For example, you take T11 residue in the unbound state, it is intrahelical, I would say, or it is in the groove, and then you can see when it is bound, it is outside. So the environment is quite different, and we believe that our molecule, since it is environment sensitive, could sense these environmental changes. So we synthesize a series of molecules, and I'm not going to go into the details, but I'll take one sequence where we have modified at the T11 position. So what we do is that first we do the fluorescence analysis, and we take a single standard G quadruplex forming sequence and hybridize, and then see what happens in different salt conditions. You can see that it shows very low fluorescence, and then in presence of KCl, which is known to form multiple structures, and mainly it forms hybrid 1 and hybrid 2 structures, and you can see an increase in fluorescence intensity. It distinguishes from duplex to one of the G quadruplex, sets of G quadruplex. And when you subject it to NaCl and it forms only antiparallel structure, you can see there is a much more enhancement in fluorescence intensity. It can distinguish, right? Same way, the tera RNA, the equivalent sequence what happens is that irrespective of the salt concentration, it is known that it forms only parallel structure. That means our fluorescence should be similar, okay, in both NaCl and KCl. So you can see in the duplex it is less, and then for the parallel structure in both NaCl and KCl, we see same structure. So you can see that our probe is nicely, or I would say faithfully, reporting the formation of different G quadruplex structures, and then we have proved it by CD, thermal melting analysis, and proton NMR spectroscopy. The same thing, of course, this is good for in vitro analysis, but then we have to see if we can generate signatures by NMR. So we form different G quadruplex structure, individual G quadruplex structures, and then we recorded the NMR. What you can see is that you can clearly see that, you know, highly resolved peaks, single peaks you get for each and every structure. And then we could also study the structural transformation in a molecular crowded environment where you form the parallel structure. The next thing what we did is that we use frogX to study the G quadruplex formation in live cell models, and essentially what had happened is that if the signatures are maintained throughout the analysis from a clear lysate to egg extract to live cells, then you can predict the structure. That's what we have done, and I'll just show one example here for an hybrid 2 structure. So it forms a signature in the intra-oocyte buffer condition, and in clear lysate also it maintains the signature peak for the hybrid structure, and in egg extract also it does. And when we micro inject and then analyze, it shows the formation of the hybrid structure, and it does not get converted into the parallel structure which has been thought about. Okay, so now we have a way using our probe to understand G quadruplex structure. The next thing what we are doing is that always the G quadruplex are not just alone, so they are associated with the C-rich region, so they can form a duplex state or a G quadruplex state or an I motif. And in order to understand these computation, you need 3 different signatures, one for duplex, one for hybrid structure, I motif structure and G quadruplex structures. And that's what we are now going about doing it, and probably if I get a chance next time I'll be able to talk about the interesting results which we are already started to get. And I would like to thank my students, and especially Arun Tanpure started with the nucleoside probe development program, and the work which I have shown is from Sudeshna. And of course, I would like to thank the generous funding from India Alliance and my collaborators Thomas Herman and Sai Krishna, and my special thanks to Jeet Kalia, who has provided the oocytes whenever we need it. And thanks for your attention.
We'll now take questions.
Minor formation of G4 across the genome.
It is possible. It is possible because the resolution we have, or the sensitivity we have with this one. So if you do with a cryo probe, then probably we might be able to capture also, which we are trying to do. We are buying it, and then we are trying to do it. But then that could be one of the limitations. But then what we have seen in the signatures of I motif and other things which I have not put it forward. Actually, we are able to even sense minor components actually, which are just 10% or something like that. We are able to capture it now.
Another question, related question is that in a system where there are normal cells and cells with physiologically different state,
okay,
is it possible for you to couple it with some trigger which releases these and then?
It's possible. It's possible. It is possible. Very much possible. So alterations, that's why I said, no, so you can have structural transformation that also be detected it. When you subjected to PEG, there was a structural transformation. It's not like, it stayed in one structure. You can have structural transformation, and by using a trigger, you can have a structural transformation, and then probably you can detect it. See, in proton NMR, you will get 8 to 10 peaks. But then, if you have 2 structures, okay, and they would overlap, it is very difficult to distinguish. But then, for each and every structure, you will get only one signal. So the peaks are resolved, so you can easily get information, I suppose.
If there aren't any more questions, we will now break for the poster session and lunch. We'll come back at 2 o'clock.